Now we are not moving to something completely different. We're actually moving to something very similar, but from a slightly different perspective. So my talk today is going to be about the coupling between hydromorphology and the biota and the complications that you will encounter trying to single out the effect of a restoration measure. And I'm an ecologist. In contrast to Christian, my background is macroinvertebrates, so you will see a lot more macroinvertebrates in my talk than fish, even though you have to believe that what I'm saying is applying to all biotic organisms in river systems. And I'm working at NIVA, the Norwegian Institute for Water Research, and I'm also affiliated to the University of Leeds. Okay, so the outline more specifically is that I want to touch upon drivers of community composition in lotic organisms, the sensitivity of biomonitoring metrics towards HYMO changes. I mean, this was also what we heard in the last lecture, but the point here is that there's a lot of methods now on the shelf that are Water Framework Directive compliant and would be natural to use in any assessment of the benefits of restoration, not least because you have to show that your measures and your program of measures are working, and some of these will be restoration. So it's very important to address this question: are they actually sensitive? Then there's the interaction between hydromorphology and other stressors that I would touch upon, the influence of confounding variables in assessing the HYMO restoration effects and how that could be addressed. And then a bit on the HYMO restoration, some examples, and how they resemble natural conditions. So that's also important in when we are assessing things and we don't get the response that we might expect from theory or from other studies. The reason could be that the restoration projects are not really mimicking a natural situation. And then I will conclude of ways to assess HYMO restorations in the end of the talk. But if we look here on this slide, we can see that we have on the local scales. This is where we are looking for effects of a restoration measure. Typically, most restorations are quite short stretches of river, sometimes only reaches. And within these reaches, you have for each organism you would have what you can call the realized niche and the fundamental niche. The fundamental is where they can live, and that's given by environmental conditions, and this is just examples, could be substrate, current, velocity, the water chemistry, oxygen levels. And the realized niche will depend upon the biotic interactions. So it might be a wonderful place to live, but if you are in the gut of a trout, you don't really have the pleasure of it, so to speak. And this is often overlooked, that of course these changes will also occur in restored sites, and it will also be influenced by the restoration measures. And this is very often the level that we look at. But on the larger scales, exactly as for the hydromorphology as such, is also controlling the conditions at the local scales by the geomorphic controls you heard about that will influence substrate conditions and flow velocities and so forth. But there's also land use history that's very important. You have dispersal. We just had the question about metacommunities, the ability of the organisms to disperse. And you have the bioregion or climate region, which is also will influence local conditions. So we need to look at this as in an integrated holistic way. Otherwise, there's a great likelihood that we'll not be able to explain what we are seeing at the local scale, and we get this mismatch between what we expect and what we actually get. But if you look at lotic organisms, streams and river organisms, there's plenty of adaptations to a life in running waters, like body shape that will reduce drag forces. That you see that in fishes, macroinvertebrates, that's exemplified here. You see it in plant species as well. The true submerged species have many sort of strategies to be really fluvial dynamic or aerodynamic and reduce drag forces. So clearly, the physical environment is a strong driver and have selectively been a strong driver in evolution. So we would expect that there's quite a close linkage between changing hydromorphology, geomorphology, and the response of the biota. So it's not only body shape, but it's also behavioral responses to a life in flowing water, life cycle strategies, what we also heard about the species traits that the species express will also reflect that they live in flowing water, and the life cycle strategies. So when do you emerge as an insect? When do you spawn? Number of fish spawn on flooded floodplains naturally, and that's all sort of linked to the hydromorphology. So we would expect that when we look at something like this, where we have a very homogenized reach, and you have something like this, which is much more complex and heterogenetic, then you would expect that there was clear differences in the biota between these two. This is sort of our basic assumption, and that you would have a higher diversity down here. And there's also quite a lot of evidence in literature, and at least in theory, that that's also the case. But there's many reasons why this is not really straightforward. So one reason is when we look at the metrics that we currently use and that are intercalibrated as part of the Water Framework Directive, we have to discuss the sensitivities of these. So this graph is showing ASPT, average score per taxon, on the y-axis, and a morphological index on the x-axis. ASPT is sort of a standard degradation metric based on macroinvertebrates. It's used in the UK, but it's also used in other member states like Finland and Sweden, and it's quite commonly used. These sites here, which is 45 smaller stream sites that varied quite a lot in hydromorphology, was really thoroughly assessed, and they were not really impacted by other stressors as such. So they were basically along the hydromorphological gradient. And this is just a normalized morphological index that was made, which goes from the very bad ones, which is close to 0, to the one that was really natural in morphology. And this was also looking at the catchment scale processes, but they were small streams. What we can see here is that there's an overall relationship. So ASPT is able to detect the hydromorphology, but the relationship is not particularly strong, given that this is the only main stressor. Another thing is also that the slope is quite shallow. So there's not, even though you go for almost 0 in morphology to almost 1 in morphology, so the widest span, you see quite a sort of a shallow slope. So it's not a really strong driver in this case, and ASPT is maybe not that sensitive, or this is at least what these results suggest. And these morphological measurements was much more detailed than what you would get in your standard assay of the methods used in many places. So that's one thing. The other way is that that's another study where we actually looked at the Danish standard metric, which is basically mainly sensitive to organic pollution, in 2 stream reaches, one that was very heterotrophic and one that was very sort of more homogeneous, channelized. And they were close to each other, so there was no dispersal barriers. Everything could be everywhere. And it was a setup of 6 streams that we compared, so 12 reaches. So quite a small study, but anyhow, I think this might be generally applicable. So the standard metric, if we looked at the DFSFI scores here, where the high score is indicating a good status and a lower score is not as good. When we use the metric as it is, then we saw that there was a difference. So the channelized stream scored lower, but that was not significant. When we took out a component of the Danish Stream Fauna Index, which is only based on diversity, and we removed the sensitivity to organic pollution out of the equation, then we got a clear significant difference. So what's actually masking this is the way that the metric was constructed. It constructed with a sensitivity element to a main stressor, that organic pollution. And organic pollution, no rocket science, is not hydromorphological degradation. There's links, but it's not the same stressor. So clearly, you couldn't expect that there would be a link. So we tried, as part of the reform project, to look at some indicators that are sensitive to hydromorphology or flow more specifically. So we have 2 types of, we use flow records for several years, and we looked in streams that had normal flow or sort of good flow conditions with, and streams that have have low flow conditions, critically low flow conditions. And this is what the two indicators are sensitive towards. And what we see here is that a high value will indicate that everything is hunky-dory and really good, and a low and a negative value will indicate there's something wrong. So, and actually what we see here is that it responds as expected, and it responds exactly as it should. So it gives good high values for the normal flow conditions, and in the streams and rivers where there are occasionally low flows or critically low flows, the scores are lower. But then if we compare now with including other metrics that are sensitive to other types of stress, like ASPT, you've seen that before, just which is an organic general, and EPT taxa, which is just the ephemeropteran, plecopteran, and trichopteran taxa, that is the stoneflies, the mayflies, and the caddis, which is also a commonly used indicator, and this indicator system SPEAR, that is sensitive to pesticides. We find that they respond almost similar to the specific tools that are out there to diagnose hydromorphology or flow conditions. So this is again normal flow, this is the low flow, critical low flow. And actually you can see the SPEAR index of pesticides is as good as the MESH index, which is an Estonian index, in detecting these flow conditions. So clearly so using a sort of a diagnostic tool might not be very helpful. I mean, you might not actually get, there might be other reasons that you get the result that you get. They seem to respond very similarly. And that's a caution, of course, you need to take, because if you apply like LIFE or MESH and think that's, I mean, because it says that this is developed for hydromorphology, and you get a response, it might as well respond to some of the other stressors as well. And more about this now. So this is from another study where we look at good habitat conditions, lower the effects of pesticides, or what is actually going on here. So we go back to the SPEAR index here. So percentage SPEAR abundance is the sensitivity indicator here on the y-axis. So when you have a higher percentage SPEAR abundance, then you have less impact of pesticides according to the system. On the x-axis, you have what is called taxonomic units. And this is a normalized, standardized measure when you have more than one pesticide. And this is multiple pesticides because this was a field study of a number of streams with multiple pesticides found in the streams. And so they were actually measured and then transferred to these toxic units that is comparable among the sites. And here you have that with increased toxic units, you have more toxicity. And these streams were similar to the others I told you about, where you had a reach that was complex and a reach that was less complex, close to each other. So the chemistry was the same, everything was the same, no dispersal barriers, and the pesticide exposure was similar as well. And it was always evident, and significantly so, that in the heterogeneous, the riffle areas, the best reach, I mean, there was always less impact of the pesticides. So there's clearly an interaction between the stressors and the physical habitat. That could be because the inverts are better to withstand impacts when they have a physical environment. But it might also be that the selection of inverts that you put into your metric system, your indicator, is not really independent. It's confounded by the habitat choice. And they are similar among these indicators. There's actually evidence that that might be the case, because it is actually the usual suspects that go through these systems to a large degree. It's the same indicator species. So that might be an issue. And that's just to say that these don't pick these off the shelf and expect that you would get a simple relationship. And this is again to the same point. And this is probably a more direct mitigation effect. We have again a number of channels. This is from an old EU project called the STAR project, and it's about 40 stream sites as I recall. So we have simple and complex channels. Again, these are independent streams. They're not within the same stream. So they are independent streams that were either complex or simple in channel morphology. And we look at low areas where there's a low total phosphorus, indicating sort of overall pollution. And there's no difference between the number of taxa when you have a low impact of phosphorus. However, if you move to the impacted sites where you have a lot of phosphorus, and probably also in many cases it would also be organic pollution, you have a clear tendency for more taxa within the more complex channels. And this is likely to be a direct effect of the more complex area provide better oxygen, more oxygen than the less complex. So the macroinvertebrates can actually resist the impact from phosphorus or pollution to a greater extent. So all this, of course, it will influence directly also on your ability to see effects of restorations. So you need to know about sort of the pollution picture, the greater pollution picture. Okay, so what's the problem in general with the methods that we have that are compliant? I think, and I have said this many times, I'll say it again, that they would develop to be sensitive to water quality, and they're still primarily the ones that are used. They focus on macroinvertebrates. The sampling method is often focused on riffle areas, specific hydromorphological areas in the stream. It's not like the one that Christian showed you. These methods that are used standardly are not, and the Water Framework Directive compliant methods are not really, they are on very small scales, normally reaches up to 50 meters, and they are often including riffle areas because this is where you have the greatest sensitivity. So if you don't have all the sensitive invertebrates in a nice riffle area, then you really sort of up shit creek fundamentally. So the sampling method is a problem, and then you have metrics. So they are based primarily on sensitivity towards oxygen concentrations, and not anything else. And hydromorphology is then, so when we compare, when we have this type of data, it's often measured on another scale. And if you use a standard method in your restoration, it would not necessarily be on the same scale as your biological sample. And then they are often very static in nature, different to what we heard about earlier today. So you have systems that don't really capture the dynamic of the system. So it's again very local scale. And that's an influence. And then you have the whole business of hydrology, which is very complicated because there's so few actual hydrological measurements out there compared to the amount of stations with biology. And for the restoration projects I've been involved in, that's always a major issue, that the hydrology is often not measured. And it's quite, as we heard and as we know, is a key player in understanding a lot of these things. So it's not difficult to see why. So we need to do something else. And what we need is to acknowledge a bit what I showed in the first slide, that we have an over-reliance on the explanatory power of the local environmental conditions. So we add more stones and we expect a response, and it might not happen. And there's the biotic interactions, which could influence the conditions and which is not normally measured. There's the dispersal, so you have the meta-community theory, as was also addressed later on. If organisms cannot disperse or can disperse in masses, you would get a masking of any effects of the habitats. You have larger scale controls on the local conditions, which is both temporal and spatial. And then you have these interactions of multiple stress, and as I just showed you, these are quite important in what you get. I mean, these things are all interacting, and you should consider that. Okay, so now we go into restoration, and my examples. I'm sorry to say, even though I'm from Norway, I'm Danish, so it will all be lowland streams, very similar to what you've seen yesterday, and that might be helpful. So we actually import stones from Norway and put them into Danish lowland streams, which is sort of a weird thing, but it's nevertheless true. So we have all these massive amount of projects, which is about spawning gravel, it's remeandering, it's floodplain restorations, and it's removal of barriers. And actually the latter is the main thing, and often very beneficial, of course. We have a real problem here, and we need to think, because clearly what we have in the toolbox might not be appropriate to detect what's there. And I think that what's really important to single out the effects of hymodegradation is that detailed measurements are necessary on the local scale as well as on the larger scales. So you cannot take something standard methods and use them necessarily if you want to be able to detect any change within your own lifetime. I mean, all of this is also about when do you want to detect any change. I mean, the weaker the method you use, the longer time it takes. So you might be dead and buried before you see any effects of what's there. But if you use a more accurate, a more precise measurement, then you might be able to detect an effect early on. That's why we want to increase sensitivity. It's not just to make it overly complicated, it's because we also want to detect a change or no change and then react upon that. So this is key, that you might have to measure more than you would normally do, and you also need to consider the larger perspective. And then, as we also heard, you need a robust statistical design, such as a before-after control impact design. It's very important to have before. Sometimes it's impossible. So you can also use a space for time substitution. I will show that later, where you compare independent rivers that have been either restored, kept as they are, channelized, or in some sort of pristine or near pristine condition, and then see how they respond to various things that you want to look at. And then I think it's very important to control for all these confounding variables in both time and space. You need to know what the confounding variables are. To show you here is that even hydromorphology takes time to improve. It's not only the biology. This is the River Gelso, which was was the oldest remeandering project in Denmark. It was remeandered in '89, and this study was done was looking at the hydromorphological conditions from 1997 and to 2008. So that's 11 and 19 years after the restoration. And this is applying the Danish standard method of habitat quality. And of course, you can accuse this for all kinds of things. But at the reed scale, it will show difference in substrates and so forth. And what we actually see here is that the quality is still increasing between 11 and 19 years. So the hydromorphology is not stable, completely stable, even after 11 years after a restoration project. So there's changing going on just in the setting of the things, whereupon we look at the biota. And seeing this is so the timescales are quite wide or long on the horizon. So that should be considered. Then it's also that what we assess as in this example, a riffle, might not be functionally a riffle in the same way. So this study was to look at two riffles that were very close together, again in a lowland stream, and they looked completely the same. So you would assess them the same if you use the Danish habitat quality index, they would be assessed as riffles and positive as such. If we looked at them in more details and look at these contour maps where we have depth, velocity, and substratum, and we measured really detailed mapping of it, you start to see that they are actually different, or one are longer than the other actually, but more than this, it's also that there seems to be maybe a little more pattern to this one. So they're clearly not completely similar, but still you would expect that they are probably the same and provide the same habitat for the biota. But if you look at a little on the bed stability, and this is the stability of how much the sediment is moved, the number of stones that were moved, and you see that and the scour, surface scour, you see that the upstream riffle is actually, most of the substrate is not moving, it's just staying there. So we labeled what was existing even under high flow events, whereas the downstream one was much more dynamic. And the same applies to when we use this penetrometer to see how embedded it was or how compact it was. So the upstream riffle was extremely compact. The downstream riffle was much more loose. So there was a hypereric zone, active zone, and interstitial spaces downstream. And you couldn't really see this before you looked at it. And what's interesting here is that you see this reflected in the biota. So this is the abundance of macroinvertebrates. This is the abundance of EPT taxa alone. And this is the particle size. And you would expect from theory and from other studies that with particle size increase, you have an increase in or numbers of species that can be there, maybe also diversity, but at least numbers because you have more surface for the animals to live in. And you see that this relationship is not existing here, so you have nice coarse substrates, but you don't get the species to match it. Whereas in downstream riffle, we get exactly the predicted relationship, very strong actually, for the total. It's a very good, strong relationship. So that was a functional, and that was a question I think was brought up earlier. What is something that is hydromorphological functioning, or it's something that will sort of, that also the biotic can respond to. So this level of detail sometimes would be necessary. And then back to the biotic. This is just something I cut out of a textbook, but we have to remember that these species respond to the flow conditions, and this is all predators, two Plecopteran predators and one Trichopteran predator, and their success in their attack success as a function of flow, and clearly they differ quite substantially in their success rate to flow. And if you change flow or you change any hydromorphology, you also change the strength of these biotic interactions. You can say in some systems it might be a key point, in other systems it might not. But you shouldn't ignore that these organisms are adapted to a life here, and they have to feed and live and whatever, and they will do it, and they will have new opportunities when you change the morphology. Then you have the upstream perturbations, and that is, of course, that we have this nestedness, so a reach down here that we restore will be influenced on everything that goes on within the catchment, and that could be agriculture that would release a lot of fine sediments that would make a lot of trouble downstream in our restored reach, where we put out all this nice Norwegian gravel. So we put out all these Norwegian stones, and what we get there is some that looks like a shitty Danish stream again. And the reason for this is not the stones that we put in. The reason is what's going on upstream. And all kind of thing will go on here. You have the loss of hyperaric zone. You have low oxygen levels. You might have scurrying at high flows for the periphyton, where you have all this is going to resuspend. And then you have the biotic interactions that change completely. You can have some predators in this system, like leeches that you wouldn't have before, and so forth. So there's this nested effect. So the catchment is very important, and also land use history, as shown by this example from Harding et al., which is an old paper and actually still one of the best showing this. That they looked at how you could explain contemporary fish and invertebrate status from land use in the 50s or land use in the 90s when the studies were done. And with the exception of invertebrate abundance, where the explanatory power was quite low, all the other fish abundance, diversity, and to a strong degree invertebrate diversity was mainly explained by land use 40 years ago. So that's another consideration. Everything might be much better now, and that's why you restore, because you improved maybe the entire catchment. But there's a legacy here, and it will take time as well. And of course, you can estimate this legacy, because for a lot of systems you would have data at least 20 years back to see the trends. And just very briefly, you have this that you can create nice habitats, so you have nice habitats, and that's what we call the habitat filter. That will the organisms will be selected through these filters depending on their niche requirements. But the key point is if they are able to get to these new habitats or biotopes. And this is often a real difficulty, of course, dispersal limitation. But the opposite is also, mass effects is also a problem. So you have really strong source population in a river, and they just colonize continuously, and it looks better than it actually is, because it looks like there is a sustaining populations, but they are not really sustaining at that reach. It's just because there is a high recruitment upstream. And of course, this needs to be taken into consideration as well. And then we have the multiple stress. So we have channelization, and we might want to restore that stream, but all the other things is going on at the same time, and which is nicely shown actually by this picture in Scotland, and you wouldn't think it was Scotland, but you can see this is the channelization is only one of several problems for this small stream, or burn as it is in Scotland. So restoration, what are we doing? In Denmark, at least, we are putting out a lot of stone, as you see here, and we put it out well sorted, so there's no real difference in stone size. But it's really sort of to mitigate some of the effects. It's lifting the bottom, and it's also to try and sort of improve the status. And this is a space for time substitution study that we did. This is a 28 different sites where you have reference on near natural, you have restored reaches, and you have channelized reaches. And this is only an analysis, an MDS plot of the substrate conditions. And what is quite evident here is that all the restored sites are very different to the channelized and the near natural sites with regard to substrate. They all separate out here, where the others mix a bit around. And this is clearly riprap structure, and this is all the addition of stone that you just saw. That of course this is not natural in any way, and it comes out quite clearly. So why is this interesting? It's interesting because it will also influence the way that we assess the biological effects. If again you look at this is a subset of the other sites where there's 6 of each, and you've got natural, restored, and channelized streams. So these are independent streams. And you look at gravel coverage with slope, and from geomorphology, you would expect a relationship between gravel coverage and slope. And for the natural stream is quite nice. So with increasing slope, you have more gravel cover. Whereas here we have the restored sites, they have much more gravel than their slope actually you would expect from their slope, indicating that the gravel was just put out, not really looking at the status. And you looked at sandy streams yesterday, and Danish streams are also very sandy. And if you look at the natural streams here, they have sand percentage from about 55 in moraine. Oh, sorry. In the more moraine to the sandy parts, where it's 90%, and the channelized streams are actually quite similar. But all the restored sites out here, they have very little sand, so they are not really restored to the natural conditions. And then sort of the clue. If you look at Shannon diversity of macroinvertebrates in relation to substrate heterogeneity, as we saw on the other side, we would expect that as the substrate gets more heterogenic, then we'll have a higher diversity. And this line is made on the natural streams, and that's exactly what you see, that you get an increase in diversity when you have naturally more heterogeneous streams. When you look at the restored sites, we don't need to deal with the channelized. When you look at the restored, they are here, so they have much lower diversity to what you would expect from the substrate heterogeneity. This indicates that you don't necessarily get the response that you are intending. So the biota might not be able to respond, and this is probably likely to be dispersal limitation, but it also might, and we also looked at redlist species, and redlist species didn't really appear in the restored reaches as well that easily. So there's these two aspects of it. And one is the ability to look at the hydromorphology from the biota, that link, and then it's the way that we're restoring. We also need to acknowledge if that's natural or not. And good restoration projects goes beyond the stream channel itself. And that's quite important because a lot of the organisms that we've looked at will have, is very closely dependent on the terrestrial ecosystem, or the riparian floodplain ecosystem. So the majority of all macroinvertebrates are insects that needs the riparian zone as adults. But fish, there's a lot of floodplain spawners among the riverine fishes. There's the highest plant diversity in the ecotone between the aquatic and terrestrial ecosystem. So clearly, I mean, that's where it's all going on to a large degree, and maybe not in the stream channel itself. This is from the Skirnor restoration, large-scale restoration, and this is all the predicted flooding, the size of the flooding, and before then there was just a straight line without any flooding. So there's really an enormous sort of influence on what was not a part of the stream before, when it was degraded, which of course used to be an even larger part of the stream early on. So a lot of things is going on here, and from a spatial point of view, it's a much larger area than the stream channel itself. So there's one thing that's of possible indicators is that the use of species traits is probably much better than identity-based methods, because the habitat, it's a habitat template theory. So species traits should directly translate into the link with the habitat. You have riparian organisms, you have ground beetles, amphibians. Christian mentioned that as well. There's a lot of things going on there, and they are likely to be respond quite sensitively, and that's also been shown. You have the whole aspect of ecosystem functioning. So we are interested to see if processes like denitrification or other types of processes are actually influenced by the restoration and not only the diversity as such, because that's an important part, and then that links very closely up to ecosystem service provision. Then, of course, alternative sampling strategies, where you use more habitat biotope mapping, is very important. And you also stratify your sampling according to this, depending on what type of restoration you have. So it actually needs to be fit for purpose. And then you need these contemporary and historic land use data at the catchment scale. You need that for understanding the geomorphology, but you also need to understand the pressure history or the history of impact of that reach that you're working in. So if we look a little more on species traits, they are closely related to the habitat template. That's the main advantage. They are less influenced by difference in biodiversity than identity-based metrics, because they don't really respond to bioregions as such. They will be across regions, so regional difference in biodiversity will not influence the output. They contain direct information between hydromorphology and the biota, as number of flow preferences is a trait. Substrate preferences, and then they link up to aspects of ecosystem functioning directly. So there are also these functional traits that would give you an idea of how is the system influenced by the restoration in functional terms. This is I will not go into detail about this, but I'll just show this is specific sets of various traits related to habitats or biotopes. I mean, the difference between habitats and biotopes is that habitats is really only one species in the core ecological sense, and you can use biotope when you have more than one species, but we just use them, interchangeable really. But that's why it's sometimes called biotopes. So you can see that in tree roots you have active aerial dispersal, isolated eggs cemented that are glued and probably to the wood. You have crawlers and shredders as mobility ways, and shredder in feeding preferences in tree roots, and they are quite distinctive. These so there's clearly something to get out of these traits. They respond to the biotopes. This is from a very large study of 8 English rivers, where they took a lot of habitat-specific sampling, not restored, but natural, sort of more natural systems. But to support my point about the functional response, this is from the 18 streams again. So this is with the 6 natural, restored, and channelized rivers, and this is the density of grazers. And so despite that we didn't see, and we actually didn't see any effect on diversity as such, we see a functional response. The amount of grazers is increasing enormously due to the amount of stones and the periphyton on the stones that are put out as part of this restoration. So functionally, these systems are changing and are not similar to the natural, and very dissimilar to the channelized stream. And one thing you could expect here is that a larger proportion, much larger, there will be a larger primary productivity that might increase biomass of invertebrates, and in the end support a bigger fish population than you would have in the channelized stream. So that could be a positive effect. But you might also have more unstable environments because you have a large accrual of algae, and you have higher fluctuation in oxygen from day to night, with lower levels of oxygen during night, because you have these primary producers that are very active during day, but in night, of course, will consume oxygen. So you don't really know, but this is bringing it all a step further and will inform much more about, be much more interesting, I think, for the people that are undertaking the restorations, especially if, as in this, if you can say that it's likely there's more fish biomass coming out of it. It's always a good message. And I will just very shortly show this, that if you look at the habitats, and Christian showed you this as well, you have the edge, the bank zone habitat, especially the bank zone habitats, and what is the backwaters. They are the ones that were really negatively affected by the regulation. And that's also the ones that we need to focus on in the restoration. And that's also where we need to look for change. In the stream channel itself, for certain for macroinvertebrates, as an example, as I know well, they are not very, if you have okay water quality, if you have good water quality, they might all be there before the restoration. There might be no sort of substantial change. But it's in these areas that the species were really lost. And especially in the backwaters, they were lost completely. And this is where the focus should be also in looking into effects. There's much higher sensitivity here than there is out here. And this is from the Gelso again, where we did the habitat mapping. And what you see here is that the various habitats where the blue is riffles, you have pools is green, you have glides, you have gravel, and then you have an edge zone. And what's quite surprising to see is that, of course, there's much more heterogeneity here where you construct a channel. And that also means that you should actually try and sample the habitats individually, and we did that. And then you get a much better impression about what's going on. And very little actually went on. They were very similar between the two reaches if it was the same type of habitat. But just one focus point here is that the edge zone here is actually much more common, or it's only existing in the restored site, where it was completely lost in the channelized reach. So it's not so much about the gravel that they put in or the nice riffle areas. Actually, the macrophytes recovered that reach really nicely, and the gravel reappeared. But the edge zone will take very long time to recover. So it could be this level of detail that are necessary, and also with focus on the important habitats. Okay, so finally the conclusions. So there's many ecological reasons, and not least human impacts, that explain why the linkage between HIMO and biota at local scales is not clear-cut. I mean, why we should almost not expect to find a relationship, in contrast to what people often believe. But this is not a real problem. It's just a challenge. It's why we are ecologists or geomorphologists or whatever. That's because we need to use our skills to deal with this problem or challenge. So standard monitoring approaches are not very likely to be sensitive in detecting HIMO changes, or at least you have to wait for years on end to see any effect. So when you are monitoring river restoration projects, you need a specifically designed monitoring program, and that's not in the projects. I mean, that's not set money away for that, but that's sort of an inherent sort of fault or deficiency of these projects. But it should, I mean, we should fight, all of us, that this is included, and it would be more costly than standard monitoring, because the likelihood of standard monitoring showing any results is so slight. So you need a robust statistical, sorry. Oh, I just need to get into that, can you, because okay, so a robust statistical design that could be BACI, or it could be a space for time substitution thing where you look at various types of streams. And then a specific habitat biotopes sampling is also really helpful. And then the confounding variables. I mean, that's often, again, overlooked. So you should look at that. Okay, that was all. Thank you very much. Okay, questions? More questions than solutions. Yeah?
These metrics that that you talk about mention light for hydrological alterations. Do you test it in lowland and upstream rivers because of maybe the predominance of mass effect on lowlands and the species sorting on upstream?
Yes. No, we didn't. In that study that I showed here was only on lowland streams. But you're right that it's likely that there is a difference in the usefulness of these indicators or systems depending on the stream type. So that could be, that could be the case. But we looked at the same types of streams here. I took it as an example because this is also the examples for the restorations, was lowland streams in my case.
The microinvertebrates, the relationship with particle size, you just see an increase in number of taxa with an increase in gravel in lowlands. Do you think again that is a mass effect predominance in lowland because of other commodities that take part of these gravels instead of in upstream that you recover, but no one can colonize it, or you have another explanation for it?
No, I don't think that's a mass effect. I think that's a true effect of the habitat filter and the lowland systems. I think it's because you can have more species when you have an hypereric zone where species that are less adapted to high flow can be in the context of the sediment. So I think that's a true effect, and I think it's an effect that you would see across river systems. So if you have an upland system and you have siltation, and compactness, and you lose the hypereric zone, you'll probably even see a stronger effect, actually a larger difference, because the lowland stream has a very shallow and not very active hypereric zone, even in riffles, whereas upland streams and more steep systems have a much deeper hypereric zone, where you potentially can have a larger diversity of species. So it might be even more pronounced when you move upstream. But I don't think there's any mass. This is not a mass effect that you see necessarily. Yeah.
And because you develop index for hydrological alterations, but do you ever relate some change in, for example, saying to someone that recover increased diversity, but do you ever relate this increase in diversity with ecosystem productivity or ecosystem resilience for you to have a demand for this recovery? I don't know, if you increase restoration and if your increase in diversity have no effect on productivity, you probably don't have a demand. So do you ever make this link to the stream productivity or diversity productivity? For example, if you increase productivity, you have a greater buffer retention capacity for nutrients, or you didn't do that yet?
No. That's what's really lacking for. Basically, that's lacking information on recovery of restoration projects and just looking at species composition. But the functional response of restorations are not very. There's only been relatively few studies using leaf litter or other things as a proxy for ecosystem functioning, where you can see effects of restoration. I also have a PhD student that is now working on the same sites now, where she looked at leaf litter decomposition and metabolism of these reaches to try and look more on the functional response. So are the decomposition rates increasing, which is you can transfer to a higher productivity maybe, or direct measurements of oxygen production, which is or respiration, which again can translate directly to productivity. So that's a really interesting question, but it's, to my knowledge, it's only been addressed in very few projects, and not many HYMO restoration projects actually that much. There's a few studies there, and it shows as an effect. But that's the next step, because as I said, a lot of these functional things is what translates directly into ecosystem services, and that's, as restoration is a human concept. It's not a natural thing. It's something that we decide as humans We want to restore something. And then there's a biological recovery from that stage, and which is dependent on the resilience of that system. And that's natural processes, but the whole restoration is not a natural; it's a social process, and that's why the link to ecosystem services is very important. I think if you want to sustain sort of the business of restoration, because if there's no effect, in the end it will not be. If there's nothing in it for us, basically, it's hard to argue for and spend a lot of money.
I think I just asked this, especially to link with stakeholders, because if you say someone that diversity increase, they probably won't listen to you. But if you probably try to relate diversity with productivity, that cannot be the case. It probably be more feasible to implement this measure because it's something that someone is interested in and paid to.
Yeah, I completely agree. Good. Coffee?
Okay.